All right, good morning and good afternoon, everyone. Welcome to GenScript webinar, Harnessing mRNA and Protein Technologies. My name is Boon Huate. I'm honored to serve as your moderator for today's sessions. We're excited to have 4 distinguished speakers joining us today from GenScript and the University of Queensland, Australia. They'll be sharing valuable insights and the latest advancement in mRNA and protein technologies. Each speaker will deliver a 15-minute presentation followed by a 5-minute Q&A sessions. We encourage you to submit your questions at any time using the Q&A box, and we will address them after each talk. At the end of the webinar, we kindly ask you to complete a short 1-minute survey. Your feedback is invaluable in helping us to continue to improve and tailor our future events to better meet your interests and needs. Now, it is my pleasure to introduce our first speaker, Dr. Alvin Chiu, Senior Protein Scientist at GenScript Biotech. Dr. Chiu earned his PhD in Structural Biology from Nanyang Technological University, Singapore. His areas of expertise include protein productions, bioengineering, biophysical characterizations, chromatography, mass spectrometry, and laboratory automations. Today, Dr. Chiu will present Turbocharge Your Biologics Design, Build, Test Cycles, a case study of bispecific antibodies. So Dr. Chiu, over to you.
Yeah, thank you, Bunhua, for your kind introduction. Very excited today to share more about this topic. So I'm sure you have all heard about like AI and the use of AI in protein design, design, build, test, learning cycles, and about how can you use like high throughput platforms in order to test like AI models. So this is pretty much the first part of my talk before I go more into bispecific antibody classification designs, challenges in producing them, and some case studies. So this is something that GenScript as a company, we are very proud and excited about. On the front page of a science cover recently by a ESM3 model, where they prove like their new model, has pretty much like the forefront in terms of protein design, and we are very proud to have tested the proteins that their model have output. So this cover, I like it a lot because pretty much we want to be like the scientists who are working on the protein design and free up our time in terms of like the production of our design. So this is where GenScript really excels at. We are constantly fine-tuning this whole design, build, and test, learn cycle at the production side, so that you can free up your time in terms of creative work here. And pretty much AI, we can, and like, it's no longer like tied down to having to do like, we can like do like de novo ab initio design, and it's no longer so much of a scientific challenge, but more of like an engineering challenge. And even in terms of like comparison with other more traditional methods like directed evolution, we could do like affinity maturation just based on various AI models. But how good it is, you still need like a good HTTP platform in order to test and find your hits for biologics work. And here's like a landscape of various tools that we have. I listed some, mostly is like open source AI-guided protein prediction, protein language models out there. And we also have like our own AI tool in-house, which I will give like a quick overview of how we try and simplify it and try to introduce this in a marketable manner, so that people with no like compute platform could also easily make use of like our whole full suite of activities. So this is what we call AI design, and it combines like pretty much 3 different tools that we have published about, from like solubility, activity, as well as the stability of enzymes in general. So I just run through a quick walkthrough of how this is still in the beta phase, so I need to hatch this, and it is mostly designed and catered towards E. coli production rather than mammalian protein production, even though I've been testing out this tool myself in-house for some of our own enzymes in mammalian protein production, and see how robust this tool whether it can work across platform. But you can register with us and let us know your interest if you would like to test it, since it's still in the beta phase, and fees are almost like very low Yeah, so very simple 5-step process. This is how the platform looks like, and we are constantly iterating it. You just create whatever like program you want. You input the sequence of interest, and so this is like the page where you can put it in, or you can upload the protein FASTA file. And here we would generate like the protein structure prediction, and all we need to input is like the number of mutations, and what property we are trying to optimize for, and the number of like mutants that we want to generate. Typically, we introduce like about 5 to 10 mutations, and then after a short time, you get like a list of hits that you can choose to directly download all the results as CSV, and you can even directly create a code and proceed to order from there. So we try and simplify the whole process from end to end here. So this is one example of how we use AI design to improve one of the protease. And here we led to, and like various screening, I think maybe about 14 candidates were screened. And then we found like, one of these candidate P9, activity and stability were enhanced. So like optimization rate, success rate is about 64%. I think it's quite respectable, I would say. Yeah. For me, of course, ideal is like you get a good scoring metrics, you get the top 5 hits, and then you screen them, you get your protein of interest, right? And here in this another case study of our high throughput platform, a lot of robotics, a lot of automation that we have in-house. Capacity in terms of this 750, I think it's more like 1,000 now. Here we screen for ESM, 178, like protein fluorescence. You're looking at like 8 business days turnaround time, small expression scale. And in this short amount of time, we can provide, like very cost-effective, rapid delivery of proving your AI model. So in terms of like the turbocharging your design, build, test, and learn cycle, right? Whatever ways that you have through your design process, be it through immunization, direct evolution, rational design through structure, through looking at structure, or just computational design. This part where we try and turbocharge that whole process, right? From build, test, and learn. And this whole workflow that you see here, pretty much is what we do in-house R&D to really turbocharge this process. And we're also trying to build like data packages such that you can export the data easily out, and so that you can analyze it back end. So sorry, I got a bit of a throat issue. So turbocharging this whole process is a bit of tongue-in-cheek for like the platform that we have in house that we call TurboTrove. Sorry. And what we do here is a lot of DOE, a lot of modeling and optimization of like from gene editing to vector to media, the whole process parameter optimization, and we have like a whole pipeline of how there's different yield and activity levels that we'd like the cell line to be performing at. So what is the performance like, right? So these are the different antibody classes, design types that we tested, and what is like the average yield in terms of HT, meaning high throughput scale, like 1 mil, 4 mil, or even 10 mils, and express level where we're starting like 30 mil onwards. So you can see that easily IgG, you're getting like really high yield of like at least 200 milligram per liter, or up to 400 milligram per liter. And recently we launched like a version 2.5 where we have like about 150% or at least 100% increase in terms of yield, and purity is generally above 90%. Now, to the topic about bispecific antibodies, for like why is it attractive and why is it such a high growth? There's like explosion in terms of demand of like many people wanting to design bispecifics, because this idea is not new, right? The key attraction is that you can bridge like effector cells and target cells by being like this bridge between them. Bring these two different targets together and achieving the functional outcome that you want. You can do standard things like what any biologics are designed to do, receptor inhibition, activation, or as a mimetic for cofactors. And out of like the top 10 selling drugs, at least top 5 of them are all biologics. And what are all the different types of design? Pretty much very varied. As you can see here, be it like a bispecific T-cell engager, we call it BiTE, basically two scFv combined together. Many different technologies that you can see here, not in whole, common light chain, cross-map. These are things that I will give like a quick run through in the next example. But we have like a growing number of approved drugs, as you can see here, over the years. So the development history of bispecific, or BSAB in short, is not new. The concept has been as early as 1960 when Nisondorf and colleagues at that time thought of this concept, right? Sorry. And this was like the first mention of the concept, but it took a very long time for this, going from this concept to the technology development, and eventually to now where we are seeing like it's becoming more applied clinically, simply because of how difficult it is to manufacture like antibodies, and how can we ensure that they are pairing in the right format, right? So this is like a very quick overview of the different technologies that were developed over time, that you can see in this flow chart. So some of the key things that happen, key events in this history is like not in whole to ensure correct pairing of the two different chains. And of course, in 99, we see like IgG4 class bispecific. 07, we see like fat arm exchange. Basically, it's just between the fat molecules and swapping the domains, basically domain swapping. And in 2012, we have like dual body, like in vitro, more like in vitro reconstitution of them. Of course, some of these technologies are patented by different companies out there. Here in our website, you see like we have listed all the different bispecific antibody designs that we have all successfully produced in-house. There are over like 100 designs, and we have like an interactive platform where you can mouse over and it will tell you things like how much, how manufacturable it is, what kind of yield are you expecting it, but it's like more like a traffic light system, low, medium, high. And high would be like what you see in terms of performance for IgG1, for example, over 400 milligrams per liter if you're going like express scale onwards. Okay, next, what kind of challenges are we looking at when we produce antibodies, bispecifics, right? So like I was saying, like light and heavy chain, they may secrete at different rate, and they are normally like on a two chain, separated as two different chains, two different plasmid when we transfect them. And then that will again lead to mispairing issue. CDR region, a lot of, sometimes, like we see like there are no issues in the sequence level. And the reason is really because of the CDR itself that causes like maybe expression problems. Imbalance in expression level, as I was talking about, and signal peptide is something that we are also optimizing in-house, so many different types of signal peptide, and how can it affect like the expression levels. Different hosts, different vectors. These are things that we constantly optimize in-house. Purification-wise, how do you separate like homodimers, for example, not or HOHO? Even if you put in some of these technologies, you still get homodimer formation in terms of like the sequence design, or they have problems binding column for whatever reason, misfolding, LC mispairing, half body, etc. And in terms of QC, how do we detect all this mispairing? For example, if you're not using a common light chain, is it mispairing to the wrong heavy chain of interest? So some case studies that I'd like to share, some of our platforms as well. So GenSmart, if you order genes from GenScript, I'm sure you heard of codon optimization using GenSmart. It's one of the market leader. A lot of companies use GenSmart as well. But in-house, we have like deep learning based like codon optimization that we call Zucue, that's only used for certain customer orders. And here you can see like the performance of Zucue codon optimization it usually like supersedes or exceeds that of GenSmart. You can see in the orange bar for both human, mouse, or rabbit antibodies tested. And some of these challenging antibodies that have almost negligible expression, you can see that Zucue indeed improve like the expression levels. Here we are looking at like heavy chain mispairing and what are some design strategies to soft heavy chain sparing. Most often than not, most people are aware of not being told, and it's no longer like it's out of patent protection. So this is not freely used in many designs, but we still get, like, for example, homodimers like this, homodimers for this particular target bispecific design, or half bodies. So these are very common byproduct, or sometimes we get like them forming unwanted, like maybe disulfide bond scrambling, and then you get really funny form of aggregations that you see the jar and you know like there's something wrong going on. And we usually like annotate them as we do our troubleshooting for our clients. Yeah. So some of the other design strategies is like electrostatic steering. We do like strand exchange, which is engineered like domain single chain, FC dimer, etc. So these are things that we will recommend to our clients as we look at the issues that happen during the production. And then in terms of light chain mispairing, so one of the most common method that a lot of people use is a common light chain in order to prevent this. If not, like here, cross map, where we swap around the domains of the variable region or the constant heavy and light region to avoid this light chain mispairing between the two different chains, or more like engineering, like here is just showing like electrostatic interactions as well between the common, between the light chain, light chain and the CH1 and the CL. Or we do like fat arm exchange, where we do like in vitro reconstitution of the two different chains. Yeah. So as I was talking about, one of the key things that we also optimize is definitely on the purification phase. So this is a single chain fat with a single chain FV or SCFV. And here in the first elution, you get in the first round of purification, you get a lot of like unwanted multimeric forms or aggregates that we want to see, can we achieve like super high purity above like 5% just with one step purification? And just by changing the column that we use and optimizing a bit of the elution condition, we could go from like 50% to 95% on SDS short analysis. And another thing that we optimize is definitely on a QC platform to tease out and identify like all the mispairing species. So here is like a reverse phase MS method that we could tease out like half body, heavy chain 1 and 2, and then where's your target of interest just at the UV detector level, and then before injecting into the mass spec, just to confirm like the different, and then what is like the ratio of mispairing. Yeah, so this is like the conclusion and what we report to our clients. And sometimes we also could do like mid-down or bottom-up MS analysis, so not just top-down intact mass. That will help in terms of seeing like more complex format that requires it, and whether there's mispairing happening and any like unwanted PTMs that happen and where which residue do they happen at. So this is like one example of how a routine intact mass analysis that we do, you couldn't see like there's a chain exchange happening between like the protein of interest. And then we do like protease digestion to figure this out between like fat 1 and 2 of this bispecific molecule. And sometimes, you know, some of our clients want more customized QC and to do like developability tests right from the start when they start scaling up to larger volume. So for example, this is like what we typically report, final yield, what's the heterodimer efficiency, and the QC we just set HPLC, SDS or CSDS and intact mass most of the time. And here we have this molecule where they want us to do a lot more like, you can definitely request and a lot more QC, like they want things that are not in the standard, right? Hydrophobic interaction chromatography, image electro-focusing for the PI, whether there's charge variance, and whether there's like subvisible particulates, thermal stability, etc. that you can see here. But one thing that stood out in this particular design is that from the image capillary isoelectric focusing experiment, we can see there are some, between these two items, there are some basic variance, okay? But the main peak of interest is still the protein of interest and some variance. Okay, quite expected, I would say. But on the hydrophobic interaction chromatography analysis, This is a standard that we run just to see one of our protein standard protein. A single peak is what we expect. We shouldn't have like a very heterogeneous species because on SDS page is a very clean peak, very clean band as well. But here we see a very obvious 2 peaks in this client's sample, and here's also to show like we have 2 different production sites in China and in Singapore, and it's very reproducible. When sometimes we'll just move things around just to see and optimize our resources. And the work that we do is very reproducible. Both picks the same pattern, and we're trying to figure out like what is causing this additional peak profile. And like you could have various examples, maybe because in the hydrophobic interaction chromatography, we use like a high cosmotropic sort, sorting out effect. Maybe it could be this sorting out effect that causes this variant. Basically, we know like there are different like this protein has different like hydrophobicity, even though it appears very homogeneous on sec. Maybe it could be glycosylation differences, oxidation, amidation state differences. We do not really know. And based on mass analysis, there is really small like peak mass changes, right? For intact mass, or when we do a reduced mass analysis. So this is like the delta mass between the peak one and peak 2. But in order to tease out, ultimately you still need to do like bottom up MS to find out which residues are being mutated, else or rather modified. Else it's very hard to tell like whether it's a mixed bag of modifications that causes this mass differences. And here is like a schematic where I break down like all the different subunits that we should be seeing, what are the different masses, where are some of the critical residues, and what could be modified. Is it oxidation at the methionine residues? Is it free thiols, or what it could be? And then we do like things like deglycosylation, forced oxidation, or forced deamidation to figure out like what is causing these 2 peaks. So long story short, we figure out that the hydrophobic variants we're looking at, or charge variants we're looking at, is due to deamidation. And by forced deamidation study, we show that it go from 2 peak to a single peak as we would expect. And so this way we do this, how like we do like cost analysis in a very systematic manner and report to our client. Yeah, so really all I'm trying to say that, you know, the key message is that we help to free up like your time to do more innovative scientific work. We offer like really industry-leading turnaround time with high yields and purity, and definitely on the back end, we do a lot more in-depth like study in order to customize to your standards that you want, and ensure like the service, whether you're producing in any one of our manufacturing site, you can expect it to be consistent in the results. So I welcome any questions that anyone may have. So this is like the end of my presentation. Thank you so much.
All right. Thank you, Dr. Chiu, for your insightful presentations. We have a couple of questions from the audience, and now we will address them one by one.
Yep.
Okay. So the first question, may I know what is GenScript protein production capacity?
Yeah, so in terms of the capacity-wise, I've got like a slide for it. So now we are rapidly expanding. Here, looking at the current, in terms of high throughput, we can go like in our China site, like 23,000 proteins per month. For Singapore site where I am based in, is 1,250 per month, and US is coming up by year end, and it will have like a huge capacity as well, as you can see here. In terms of like scaling up above like 30 mils, so this is like the numbers that you're looking at. Yeah.
Okay, great. And how does the purification strategy differ between conventional monoclonal antibodies and bispecific antibodies?
So a lot of times, the performance that we show is on like monoclonal antibodies, and you get like a much higher yield from a smaller volume. And when we talk about bispecific, we typically recommend at least 2-step purification, and a higher starting volume of like at least 30 mils to 100 mils, depending on how complex the molecule design is. So it's usually require like at least a 2-step purification.
Okay, we are taking one more question. Regarding the AI design you mentioned, does
Mhm.
it work on antibody design or mammalian expressions, or only in E. coli?
Yep. So right now the AI design is really designed or rather trained for enzymes and in E. coli production. But that is not saying that it wouldn't. I'm not ruling it out. Like it cannot work in mammalian expression, but it's not designed for that. Or neither is it designed for antibody production. Yeah. But I shared like the whole table. There are many tools out there that are specifically designed for antibody design. Yeah.
All right, got it.
Yeah. Open source.
Um, yeah. Um, all right, I think there's no more questions. So thank you once again, Dr. Chiu, for the engaging discussion and thoughtful responses. Now let's move on to our next speaker. It is my pleasure to introduce Dr. Christian Nefsker, Group Leader of the Reprogramming Aging Laboratory at the Institute of Molecular Science at University of Queensland, Australia. Dr. Nefsker holds a master's from the Technical University of Munich, Germany, and a PhD from Monash Institute of Pharmaceutical Sciences in Australia. He is an expert in reprogramming, epigenetics, and cellular aging with more than 4,500 citations and publications in renowned journals of Cell, Nature, and Science families. Today, Dr. Nefsker will be presenting a developmental mechanism gone awry, AP1 link chromatin opening as driver of organismal maturation and aging. Dr. Nefsker, the floor is yours.
Thank you so much for the kind introduction, and also thank you for the invitation. It's much appreciated. So today I'll be presenting about my lab's work related to organismal maturation and aging, and I want to make a disclaimer up front that I only have literally one slide about the use of RNA technology, but I think it's quite critical for the future of my lab's research. So there is mounting evidence that the key driver that facilitates our bodies to age are based in changes in gene regulation or the epigenome, rather than irreversible genetic mutations that has been believed until perhaps a decade ago or so. And even though we more or less convincingly know that it is changes in the epigenome that drive aging, what the nature of these changes is still an ongoing question. Is it random changes as proposed by David Sinclair and his work, or is it potentially an ordered targetable process with a developmental link? Considering that many of the phenotypes that we link to aging, like immune senescence or regenerative decline, already play themselves out or start well before we reach adulthood, because regenerative capacity is highest very early in life, as well as COVID has taught us about immune cell function, which peaks in children. And my lab is very interested in trying to understand the role of transcription factors in these processes, both organismal maturation as well as aging, because transcription factors are master regulators of cell identity and also have considerable control over the epigenome. As you know, all cell types in our body have the same DNA, but different aspects of it are active. And depending on their functional requirements, and transcription factors control that. There are, for example, different combinations of transcription factors that are active in brain cells, nerve cells, to drive nerve cell-specific gene expression programs, whereas there are other ones in hepatocytes to activate the genes required in these cell types. And understanding what combinations of transcription factors underpin what cell state comes with the power to manipulate, as has been shown quite impressively by Shinya Yamanaka's discovery, for which he got a Nobel Prize, where he showed that by overexpressing 4 transcription factors that are linked to the early embryonic cell state, OCT4, SOX2, KLF4, and c-MYC, one can reprogram both adult cells as well as aged cells all the way back towards the pluripotent state. And then these resulting pluripotent stem cells can be used to generate new animals, secondary animals, via tetraploid and chimera assays. And these secondary animals that are derived from these initially aged cells have a normal lifespan and they're not displaying any premature aging phenotypes. That is another evidence that by reformatting the epigenome, which reprogramming to pluripotency does, aging phenotypes are erased. So my lab is interested in trying to understand whether there are transcription factors that drive these processes forward and whether these transcription factors are potentially conserved across cell types so that we can come up with strategies to dampen the activity of these transcription factors to slow down age-related decline or potentially effect partial restoration. And in order to study that, for a study that was published last year in Cell Metabolism, we profiled 22 cell types at high depth, isolated from young and aged animals, for which we performed ATAC-seq and RNA-seq, and we complemented that with single cell validations. And to study developmental processes, we reanalyzed published data sets for 29 mouse and human cell types and their maturation processes. In most cases, it was fetal versus adult cell states. But we also had continuous data sets that were spanning the entire continuum all the way from fetal to aged, so that we can understand transcription factors drive remodeling. And our study showed that indeed it's an actually quite ordered process, both during development and then in aging. And conceptually, what we found was that when the different cell types that make up our bodies arise, and there are around 300 during fetal development, cell type specific combination of cell identity transcription factors commission the activity of developmental genes that are required for growth. And adult genes are yet switched off at this stage of development because their gene regulatory elements are hypermethylated and they have less binding sites for these cell identity transcription factors. However, they make up for that by having more binding sites for the AP1 transcription factor, which is induced in response to a range of stimulus processes, and this is the case across cell types. And we found that rising activity of AP1 during development together with hormonal cues is breaking open these adult gene regulatory elements together with a loss of DNA methylation in these regions. And that in turn triggers shutdown of these developmental genes to slow down growth processes. And that is happening via two mechanisms. For once, AP1 linked chromatin opening triggers a mild downregulation of the expression of these cell identity transcription factors. And secondly, there's AP1 exposes competing binding sites for AP1 cofactors that are present at these developmental gene regulatory elements. So then getting redistributed to these adult gene regulatory elements. Unfortunately, or potentially fortunately, we found that a process does not stop once our bodies hit adulthood. There is ongoing AP1 linked chromatin opening because AP1 is also induced in response to stress and inflammatory stimuli, which further opens chromatin and leads to a further destabilization of the activity of cell identity transcription factor commissioned gene regulatory elements that drive the expression of useful genes. We also validated that there is transcriptional upregulation of a number of AP1 subunits in aging across cell types, linked with a mild downregulation of the expression of cell type specific cell identity transcription factors, as I've mentioned. And I don't want to go too much into this figure, but what we're showing here is each of the columns here on the left side represents chromatin regions that open or close for one of the 22 adult versus aged cell types, so 17 fetal versus adult cell types that we looked at. And what we found is that, also as I mentioned, regions that open as part of both processes overrepresent AP1 binding sites, but they have a relatively similar cofactor profile to allow these redistribution events. And as part of our published work, we profiled and characterized key cofactors that AP1 is dragging into these progressively opening regions. And as Emmanuel Serrano wrote in a preview article for our publication, AP1 steals the show in chromatin aging, quite literally. We validated these processes via a number of assays, cut and run, for transcription factor binding shifts. We also show that this chromatin reconfiguration underpins age-related transcriptional changes, and there is a progressive ongoing shutdown of developmental genes, even in aging, while there's conversely hyperactivation of stress and inflammatory gene signatures. And we further validated that AP1 is the key driver, which is visualized here for 3 different cell types. So I was showing that the regions that open as part of aging overrepresent AP1 binding motifs. They are over-enriched there, or whereas the regions that close overrepresent cell type-specific cell density transcription factors, including T transcription factors for fibroblasts. So when we overexpress AP1 subunits in vitro in fetal fibroblasts, we can trigger AP1 linked chromatin opening as would be expected. And the regions that open in response to AP1's pioneer factor activity, of course, overrepresent AP1 motifs. And this triggers a closure of TEAD1 overrepresenting regulatory elements according to our model. And this also drives in vitro premature aging phenotypes by triggering premature senescence in these fetal fibroblasts. We also show, which is probably more relevant for a human aging, that stimulus and stress processes can also trigger these kinds of rearrangements that we link to aging by triggering AP1 upregulation. And we validated the important role of progressive epigenetic derepression by showing that if we deplete a repressive histone modification that's linked to facultative heterochromatin, H3K27ME3, via PSC2 inhibition, that we can also trigger these kinds of rearrangements akin to what we get when we overexpress AP1 itself. And it's not just loss of repressive histone modifications that's enabling this progressive activation of late-life regulatory elements through AP1. It's also loss of other layers of gene repression, like DNA methylation. What you can see here for hepatocytes is that on the x-axis, you can see chromatin accessibility changes. So regions that open as part of liver aging in the young state, which you can see on the y-axis, have very high levels of DNA methylation. However, these DNA methylation levels erode in response to rising AP1 activity. So it seems that there's a synergy between rising AP1 activity and loss of epigenetic repression to drive initially the developmental program, and then beyond that, age-related decline. So one of the things that my lab is now interested is trying to understand how this rather predictable chromatin remodeling manifests at the tissue level. And the liver is a very lovely organ system to study this, because it has a very ordered structure. Hepatocytes that are rowed up between blood vessels that provide and drain the organ nutrients. These hepatocytes have or fulfill different functions, and these functional separations are encoded to some degree, at least via growth factor gradients that emanate from the central vein, Wnt signaling, and arrest signaling emanating from the portal vein, which again endows them with different metabolic roles and functions. And in order to understand how the chromatin remodeling manifests at the tissue level, we performed spatial transcriptomics for liver sections from young and aged animals. And we then wrote up the resulting spatial dots along the periportal to pericentral axis. And we found that in young livers, there is the expected zonation pattern. Periportal hepatocytes express periportal marker genes, whereas pericentral hepatocytes express pericentral marker genes. However, this division of labor decays in aging. The periportal signature disappears from middle age onward already, actually. However, the pericentral identity is largely intact. And aligned with this, we did not find any alterations in Wnt signaling, but we found that a key potentiator of REST signaling, EGFR receptor, was downregulated from middle age onward. And to link this to our chromatin modeling, we also looked at gene regulatory elements that control the expression of EGFR. And we find that they are progressively shutting down as part of aging. So this provides a model to explain how predictable chromatin remodeling can also lead to predictable changes at the tissue level. And this is important for the liver as well as for a host of other organ systems, because age is the biggest risk factor for most degenerative diseases. In the liver, we proposed that this kind of metabolic remodeling predisposes for metabolic liver disease. Evidence that aging is driving age-related diseases is, for example, provided by people that suffer premature aging syndromes. They manifest age-related diseases much earlier in life than others. And the reverse is true for dwarf mice, for example, which live considerably longer than normal mice. They have delayed occurrence of age-related disorders. So we postulate that if we reignite more useful capacity and regenerative capacity, useful capacity and regenerative potential, we may be able to come up with therapeutic interventions to either prevent or potentially treat age-related disorders. And our model provides a number of intervention angles for that. The obvious one, which I'm not speaking about today, is to dampen and reduce AP1 activity. Another angle that we're working on with RNA technology is to reinforce transcription factors with reduced activity that are linked to useful gene regulatory elements that lose activity. And while most of these transcription factors, as I mentioned, are cell type specific, there are some that are shared across cell types. And that's the most prominent ones here are NFI transcription factors with known roles in development that are linked to these useful developmental regions across cell types. And we're using RNA technology, in vitro transcribed mRNA with modified bases via the clean cap strategy to overexpress NFI transcription factors, for example, in presenescent fibroblasts, which allows us to reignite more useful proliferative potential. And we did the same for T cells that we isolated from aged animals, which also have very pronounced proliferation defects. And likewise here, NFI overexpression increases or reinstates more useful cell proliferation. Of course, these are very superficial characterizations, and we're trying to go more in depth and also perform in vivo studies with the liver a early target, because it can be reached quite easily via mRNA through tail vial injections. So to summarize what I've shown you, basically the lab created a model for transcription factor network remodeling during maturation and aging. And we've shown that AP1 link chromatin opening initially progressively activates adult genes during maturation of all our different cell types together with hormonal cofactors. And this in turn has a developmental role to destabilize cell-ended transcription factor-rich developmental gene regulatory elements, to progressively reprogram our cells and tissues towards adult tissue function. Unfortunately, this developmental mechanism goes awry beyond adulthood because there is ongoing AP1-linked chromatin opening through continued AP1 causing stimuli like inflammation or stress, which leads to a further shutdown or an active predictable shutdown of useful gene regulatory elements. And we propose that this rather predictable mechanism is underpinning many phenotypes linked to aging. And we've shown that at least at the transcription factor network level, age-related molecular remodeling is not a random process. And this gives hope for new avenues to mitigate age-related decline and treat age-related disorders, namely by inhibiting or dampening the activity of the driver transcription factor family, or by reinforcing developmental or cell-ended transcription factors in a fashion to also reignite more useful capacity by resupplementing these transcription factors that have become deficient. And rather than, again, aging being a random process, our study suggests that it's more akin to a maturation-like process. It's not exactly the same because there is no involvement of hormonal cues, but it shares many attributes with the process that drive development. That just leaves me to thank the people in my lab that were involved in this project. Ralph Patrick and Marina Naval-Sanchez drove the computational analyses, whereas Xiaoli Chen and others in the lab drove the wet lab work. I also want to thank all the collaborating groups, which are listed on the right-hand side, that helped with the cell isolations using their established protocols. I would like to thank ongoing immunology collaborator suite or DU's group, as well as our liver collaborators, Elizabeth Powell and Kate Irvine, together with Kwan Agun, who supported the spatial transcriptomics work. And I would also like to thank the funding bodies, as well as, of course, GenScript for the opportunity to present here today. Thank you very much for your attention.
All right. Thank you, Dr. Netsker, for your fascinating presentations. We have a few questions from the floor, and let's take a moment to address them. You may also refer to the Q&A box for the question asked. So we have the first question. Do you think intake of metil-dominant diet, such as high folate, choline, is a good idea for the elderly?
That's an interesting question, considering that there is loss of repressive histone modifications as well as DNA methylation. It's possible, but I don't want to make concrete recommendations. It is possible that it could slow down this decline of epigenetic repression, which is one of the two aspects that is driving this progressive opening and destabilization of the cell identity network.
All right, thank you. And is there any possible that AP1 was coincidentally happened together with an unknown factor, because AP1 is a quiet universal TF for a lot of genes?
Yes, also an excellent question. So AP1 is in a way the conserved transcription factor across cell types, but it has cofactors. So even though, and this is part of ongoing work, so there are many inflammatory cofactors that, like STATs, NF-kappas, betas, IRF transcription factors that help facilitate activation of stimulus-specific stress processes that work in synergy with AP1. So that's one of the cofactors. And the other one is, even though cell anti-transcription factor binding sites are overrepresented in the developmental genes, we still find evidence for the presence of cell anti-transcription factor binding sites in the regions that open as part of development and aging. They are just less abundant and they have lower affinity binding motifs. So we think that that's another mechanism that keeps these adult gene regulatory elements sealed off. So they have not only less gene regulatory, not less binding sites for cell type specific transcription factors, but also reduced binding affinity so that they cannot get opened at the start of life. But we think that these cofactor profiles are important to allow cell type specific opening in synergy with AP1. I hope that wasn't too detailed.
Yeah, thank you for your answer. And we're taking one more question. Was any experiment done to see if reversal of AP1 or any other age progressive transcription pathways can delay or mitigate cell aging?
Yeah, so yes, we are now working on AP1 and it published data, so I don't want to go too much into it, but it's looking promising. There have been studies a long time ago already with a key AP1 cofactor, NF-kappa beta, and there were studies, I think it was Adler et al., maybe 2012 or 2010, that inducibly removed NF-kappa beta from aged skin, and they saw some restoration. So yes, blocking AP1 or its key cofactors seems to be a quite viable strategy.
All right. Thank you once again, Dr. Nevsker, for the insightful discussions. Thank you.
Thank you very much for your.
Yes. So now, moving on to our next speaker, it is my pleasure to introduce Associate Professor Jason Lee, Principal Research Fellow at the University of Queensland, Australia. Associate Professor Lee leads the Epigenetics and Precision Medicine Laboratory at the Fraser Institute and also holds adjunct positions at the Queensland University of Technology and Griffith University. He completed his PhD at the University of Sydney and pursued postdoctoral research at the Dana-Farber Cancer Institute, Harvard Medical School, focusing on nuclear receptors in breast cancer. His current research spans molecular epigenetics, hypoxic signaling, and cancer metastasis, alongside a strong focus on drug development and biomarker discovery of cancer therapy. Today, Associate Professor Lee will be presenting on epigenetics regulation in cancer, multi-omics approach. Associate Professor Lee, the floor is yours. You're on mute.
Okay, can you all see the screen?
Yes.
Okay, all right. Thank you very much for the opportunity to actually spend some time going through some of the approaches that we've been using in our lab in looking at the cancer, focusing on a couple of epigenetic enzyme inhibitors in the lab. So hopefully at the end of the sort of my presentation today, it will be really sort of kind of, it will be nice to sort of kind of get some feedback as well as, instill some approaches that you can take away with. So, Christian's already kind of given a nice background of how epigenetics is actually important in terms of aging. And as you can see over here, as the caterpillar matures, it becomes a butterfly, has the same DNA, but different phenotype. So, it also applies to a lot of the cancer settings where, the genetic makeup of the primary tumor versus the ones that are resistant to treatment or metastatic tumors tend to have the same DNA or mutation profile, but it's the epigenetic changes that allow them to become more aggressive, and hence the importance of looking at epigenetic regulation. So about 15 years ago, Time magazine had actually profiled an article talking about it's not just the DNA that dictates your future. There is other things involving epigenetics that would change the destiny of your being, and also for the cellular level. Another way to think about epigenetics is to actually think about it in terms of the punctuation. So, have the same letters. If you could think of, all these letters in that sequence is like the DNA, but depending on where you put the comma, you could actually change the meaning drastically. So, if there is anything that you take away with after my talk, if you could think of epigenetics as a punctuation mark, and, that'll be really good. So in terms of the molecular level, as Christian sort of talked about the whole chromatin remodeling, in the relaxed formation of the DNA or the chromatin, you have the exposed DNA portion that is accessible by the transcription factors to turn on gene expression. And on the right, you've got the compacted or the condensed chromatin that doesn't allow the transcription factors to actually bind. So that the gene is actually not expressed. And as Christian sort of kind of mentioned that, there are some histone modifications and also AP1 complex proteins that sort of, control the expression of genes. My lab focuses on enzymes that play a role in histone modification and hence the structure of the chromatin itself. And two examples are, gRNA or euchromatic histomethyltransferase 2, which there is a 2 compound that we could inhibit the enzyme activity using the UNC0642. And as mentioned by Christian, also this is the PRC2 component, EZH2, that a couple of the drugs are in fact approved for treatment of some cancer patients. So these two enzymes methylate the histones and lead to the silencing of some tumor suppressive genes. And our approach is to go and inhibit these enzymes to make sure that these tumor suppressive genes are re-expressed. So overall in my lab, we've been dealing with quite a lot of different cancer types, but the problem in the cancer setting is the recurrence. It's the therapy-resistant cancer that really is the problem for treating cancer patients. So the idea that we're trying to develop in our lab is to identify a biomarker that can allow us to go and target which epigenetic modifiers and perhaps the target genes to combine with some of the treatments that are already used to treat these cancer patients to turn these aggressive phenotype into a more sensitized tumor setting so that effective tumor cell killing can be achieved. So this is a little diagram that I actually stole from my honor student, Damien, who's actually working on circular RNAs, which I'll talk about in the later part of my talk. But obviously, you've got DNA methylation that is, well characterized. We focused on histone modifications, but there are also non-coding RNAs play a role together to control gene expression and also changing the phenotype of the cancer cells. And I would like to actually mention a few different things that we've been using. We haven't used all of these, multi-omics platforms, but it's nice to see that the technology has actually come together, that we can utilize some of these things together to gain more meaningful information going forward. So in terms of the genetics, so if you go to the TCGA data set, you're looking at quite a different types of cancer patients. And this is just an example of gRNA or EHMT2 histomethyltransferase copy number gain and amplification. So across the different types of solid tumors, we can actually see, between 40 to 50% of my patients harbor copy number gain, which translates to the tumor cells having high expression of the RNA transcript, going from melanoma, ovarian, hepatocellular carcinoma, and also, breast and colorectal. So this is something that is actually happening across all the different solid tumors. And we thought that it might be an idea to go and inhibit this enzyme because it's known that some of the tumor suppressor genes are turned off in cancer setting due to the increased histomethylation. And we've been using this compound called UNC0642 as an enzymatic inhibitor of G9A histomethyltransferase. And so when the chromatin is in the condensed format for silencing some of the tumor suppressors, when we use the inhibitor to block the activity of the enzyme, we can unwind the chromatin so that the transcription factors can come and bind to activate its target genes. So that is the model that we're using, and we've applied this inhibitor treatment in a high-grade ovarian cancer model where we utilize the patient-derived xenografts to look at the efficacy of chemotherapy, namely carboplatin over here, and also in combination with our G9A inhibitor. So as you can see on the left, ovarian cancers actually grow in nodules. So the number of nodules represent the tumor burden in these mice. And the combination obviously shows the lowest, not too significant compared to the carboplatin alone, but in terms of the tumor weight was also down. And on the right, if you look at the survival of these mice that are bearing these tumors, is significantly increased in the combination of carboplatin and G9 inhibitor compared to the single drug treatments. So it seems like we can sensitize these tumors to chemotherapy, and the biggest implication of this work is the fact that we can achieve better chemotherapy response when you combine with G9 inhibitor, therefore allows us to utilize less or lower dose of chemotherapy, and which will be beneficial for the patients because it reduces the side effects. As you know, in cancer therapy, we are going away from treating patients with chemotherapy towards more targeted therapy. And this is a collaboration that we've actually done some work using chemo-resistant PDX models with Claire Scott down in Weihai. And it seems like the UNC0642 is performing somewhat okay compared to the olaparib in chemo-resistant model. And again, the survival of these mice are going beyond 80 to 100 days, which is much longer than the untreated mice group. So we've actually established that the drug can be used in conjunction with chemotherapy and quite efficacious in terms of controlling tumor burden in mice. But what is really happening at the cellular level? So we utilized a whole monitor imaging to see what the drug is actually doing. So on the left, you just saw all the cells that are growing and dividing and moving in the vehicle treated. And on the right, the G9A inhibitor treatment really stops the cells dividing and also moving about. And we've quantified that in terms of the distance that these cells are actually moving. And as you can see, the G9A inhibitor treated cells actually move less. And we've applied that in a hypoxic model as well. So if you see on the right, on the bottom there, if you put cells into hypoxia, all the cells are moving about much better. So this is the notion that a hypoxic microenvironment allows the tumor cells to escape that primary site for metastasis. And in the same hypoxic condition with G9A knockdown using a hairpin mediated knockdown, as you can see that the motility of these cells is drastically reduced. So suggesting that perhaps the inhibition of G9A might be applicable for modulating or inhibiting metastatic potential of these cancer cells. So what next? So in ovarian cancer, there was about 46% of the patients in the TCGA data set that would benefit from G9A inhibition. But also, if you look at PRC2, EZH2, which is another methyltransferase that is known to silence tumor suppressor genes is about the same proportion of patients harboring copy number gain or amplification. So we thought perhaps we might be able to utilize co-targeting both enzymes using a small molecule inhibitor. And so if you add up, the proportion of patients that can benefit from both the HMT2, which is the gene that encodes for the gRNA protein, and EZH2, it's going up to about 70%. So which is a decent number of patients that can benefit. So we've looked at the expression of the transcripts in the tumor versus the normal. As you can see, both the HMT2 and EZH2 are highly expressed. And if you look at ovarian cancer patients that have undergone debulking surgery, where they try to actually take the tumor, and you can see that if the patient hadn't actually gone through an optimal debulking, the progression-free survival of these patients is actually quite bad. So the idea is, we're going to target both histone H3K9 and histone H3K27 methylation to see what really is actually happening there. We've performed some RNA sequencing analysis to find out, what it is that is actually happening with the combination. And as you can see that the combination, the UNC0642 and the GSK compound that inhibits EZH2, as you can see, there are a number of genes that were hyperactivated compared to a single drug condition. And on top of the list, we actually found this cytokine called interleukin-24 appears to be robustly upregulated, and as you can see, the transcript level of the combination treatment at 48 hours is actually going up to about 50 to 60-fold. And what was amazing about this work was the fact that IL-24 has been identified as a factor that drives tumor cell specific death, but not affecting the normal cells. And obviously, we've done some chip chromatin IP experiments to see that the inhibitors are actually working on the IL-24 promoter. So both K9 dimethylation and H3K27 trimethylation was actually down. And the demethylation of these histones was associated with increased in POL2 recruitment, which represents that this gene is actually actively transcribed. We've done some overexpression of IL-24 without using an inhibitor, just to be able to show that, well, can you overexpress IL-24 to induce cell death? And as you can see across the different cancer cell types, going from triple-negative breast cancers, ovarian, and melanoma, and you can induce cell death by overexpressing IL-24. And we did a rescue experiment where we switched off IL-24 using DCAS9 CRAB complex, where you deliver this transcriptional repressor complex. So even when you treat these cells with our inhibitors, you can no longer activate IL-24. And as you can see, these cells don't die in response to our drugs when you silence IL-24. So that was all good. In terms of the aspect of targeting two epigenetic enzymes. Some of the things that we are interested in at the moment is looking at the drug distribution. I mean, we can assume that the tumor is actually treated with the drug, and so what we want to demonstrate is how the drug is actually distributed around the tumor. And we're doing some MALDI metabolic analysis to be able to actually see where the drug is, and then correlating that to the metabolites, which you can compare between the drug-enriched and the depleted regions. One thing that we are ongoing, I've actually received this data from Jessica Ma's group just last night. We're using this Stomic stereo-seq, which allows us to look at the whole transcriptome, non-coding RNA at single cell resolution. And we've got two regions in the drug-treated tumor on the right. So you've got, the blue region. One is the drug-enriched region, and then you've got drug-depleted region, which is marked as number 2 in red. And if you can actually look at the transcript differences, and we start to see some of the genes that are upregulated, and also compared to the DMSO vehicle-treated tumors, we actually get a profile of genes that are specifically regulated in that manner. So now, we're not only looking at, spatial transcriptomics in a tissue to look at the cell types or enrichment of certain genes, but we can now overlay that with what the drug is actually doing within the tumor. We've done some collaboration with Peter Sorgerslap at Harvard Medical School looking at ovarian cancer in the pre-lesion groups. And just to summarize that, we were able to identify that EHMT2 copy number gain was actually happening at the non-cancerous setting, which means that the copy number gain or the amplification of this region is an early event. Which was a new finding for us in looking at the progression of ovarian cancer as well. Just a couple of minutes at the end of my presentation, we're working on a circular RNA, and we're trying to actually work that as a novel biomarker for early detection of ovarian cancer, but also utilizing the peptides that are actually made from the circular RNA for vaccine development. We're using some nanopore long-read sequencing to identify circular RNA that is specifically up or down regulated in the ovarian cancer compared to normal. And we're finding out that the circular RNA expression is controlling the chemo sensitivity in some of the ovarian cancer cell lines. And what's really cool about circular RNA is the fact that you can use digital PCR to actually get a quantification of how these circular RNAs are expressed. And one of the things that we are actively working on is to use liquid biopsy method to identify to measure the circular RNA levels in patients that have low grade or high grade serous ovarian cancer, but not detected in the benign or the control healthy plasma samples. So we're quite excited about this finding, and we're hoping to extend the number of samples that we could examine for a test development. Just as a last thing, where I mentioned circular RNA derived peptides, and we think that there are some peptides that are being derived from circular RNA, and because these are derived from the circular RNA that goes through a novel open reading frame, it creates novel peptide sequences, and hopefully we can use some bioinformatic analysis and screening tools to be able to identify which can be used as a neoantigen. So, we start from genomics. We look at CHIP and ATAC sequencing to look at the chromatin structure. Obviously, we need to look at the transcriptomics to make sure which genes are going up and down. Something that I haven't actually mentioned today, but obviously, the two enzymes that we are working on are involved in methylation of proteins. And perhaps we can marry that with metabolomics and also looking at the phenotypes that we get in response to the drug. With that, I would like to actually thank the funders, the donation from patients and consumers that have given us a lot of blood and cancer samples, and also the past and present members of my lab. Thank you.
Hi, thank you, Professor Lee, for your excellent presentations. And again, to all the attendees out there, if you have any questions, feel free to drop them at the Q&A box and we'll address them. It seems like everything was crystal clear. No questions at this time. Yeah, so that's a great sign of a well-delivered presentation. Thank you again, Professor Lee, for sharing your expertise. If any questions comes up after the session, feel free to reach out to us directly. And now we would like to move on to our final speaker today. And it is my pleasure to introduce Dr. Yuxiao Liu, Senior mRNA Synthetic Scientist at GenScript Biotech. Dr. Liu earned her PhD in biomedical engineering from Southeast University in 2021, specializing in capillary microfluidics and hydrogel microcarriers. She has published multiple papers in leading scientific journals, including a review in Chemical Reviews. After her PhD, she focused on advancing mRNA vaccine delivery at One Globe Medical. Now at GenScript, Dr. Liu leads the development of lipid nanoparticle LNP formulations for mRNA delivery, supporting innovations in gene editing and targeted therapeutic delivery. Today, Dr. Liu will be presenting on LNP-enabled mRNA technologies, applications in vaccines and beyond. Dr. Liu, the floor is yours.
Okay, thank you for the introduction. Can you see my screen now?
Yes, we can see it.
Okay, thank you. So today I'm really glad to have this chance to share some insights about how LNP can help mRNA work better, not only in vaccines, but also in other very exciting fields. Well, before we dive in, let's have a quick look at the history of mRNA technology development. From the early discoveries in 1960s to the success of COVID-19 vaccines in 2022, the mRNA technology has grown into a very powerful and versatile tool. And after decades of development, mRNA technology has become a real therapeutic platform. The most commonly known application case for mRNA technology is COVID-19 vaccines. But nowadays, the application field of mRNA technology is far beyond vaccines. It can be used for cancer vaccine, cell therapy, genome editing, antibody production, and protein replacement, etc. To realize all these kind of applications, the delivery system has played a very important role. When talking about delivery system, the lipid nanoparticle, or LNP for short, is a very important example. So before we dive in, let's have a quick look about the general structure of lipid particles. So it usually consists of four main components. The first component is the ionizable lipid, or the cationic lipid, which can self-assemble with nucleases or other payloads like mRNA, siRNA, or DNA plasmids, etc. And the second component is phospholipids, or we can say helper lipid as well. It usually used for maintaining the structure and stability of the LNP particle. And the third one is cholesterol. Cholesterol is used for the structural lipid and could maintain the stability and structure of the LNP particle as well. And we also have the PG lipids, which can promote the circulation of LNP particle, especially in vivo. So in addition to general LNP formulations, nowadays we also have various types of targeted LNP for enhanced RNA delivery. When talking about targeted LNP, usually it can be divided into three main categories. The first one is active targeting LNP. Usually, to making this kind of targeted LNP, we can conjugate specific ligands onto the surface of the general LNP, like peptide, antibody, or some specific small molecular that can bind to specific receptors. And the second type is passive targeting LNP. For this type of LNP, it can be achieved by adjusting the physical and chemical properties of the LNP, like size and surface charge. And the third type is material-based targeting. From my point of view, it can be also involved in the passive targeting LNP, since it's usually achieved by adjusting the components or using novel ionizable lipids to achieve the specific biodistribution in vivo. And it's kind of a strategy to by attuning the properties of chemical and physical and chemical properties of the LNP. So in GenScript, we also offer various kinds of targeting LNP. As you can see, we offer active targeted LNP like CD3 antibody conjugated LNP for T cell targeting, CD31 antibody LNP for stem cell targeting, and also GalNAc LNP for liver targeting. We also have some passive targeted LNP like 20-DOTAP LNP, 18-PA LNP, and 50-DOTAP, which is made of 5-component LNP and is specific for liver targeting, spleen targeting, lung targeting. And also we have developed 3-component LNP also for lung targeting. And in these slides, I just want to discuss a little bit more about the different mRNA formats that can work with LNP formulations. The first one is linear mRNA, which is the most widely used. And if you are just the newly entered to the mRNA field, or you want to conduct some proof of concept studies, the linear mRNA may be more suitable for you. And for linear mRNA, it usually has strong protein expression, but the half-life is relatively shorter and it's mostly common. And the second type is self-amplifying RNA, or the saRNA for short. It have a special structure in their sequence, which called the replicase gene structure. And that means it can self-replicate it in cells by themselves. Since it can self-replicate it, it has the strongest protein expression level and the expression duration at the longest among these three types of mRNA. And usually it has relatively high immunogenicity, and the saRNA is more suitable for applications in the vaccine fields. And the last type of mRNA is circular RNA. It doesn't have free end, and due to its structure, its circular structure, the stability of this type of mRNA is relatively high compared with the linear format mRNA. And usually it have longer expression duration. And for circular RNA, it is very welcomed in those cell therapy application fields. In GenScript, we just offer a very versatile, a very flexible LNP platform that can work with all these three types of mRNA. Well, in next part, I would like to share some real internal case study from GenScript. And the first one is about using the self-amplifying RNA to develop COVID-19 vaccines. As you can see in this case study, we use the SM102 LNP to encapsulate the SARS-CoV-2 RBD Delta mRNA. And we found that it can exert very strong and prolonged serum titer. Even after 90 days, the serum titer just remain a very high level. And this case study just show the potential of saRNA in developing vaccines due to its prolonged expressions. And regarding the saRNA, we also find a very interesting phenomenon. That is, we found that the saRNA itself might have some intrinsic tissue targeting properties. That means when we use the same LNP formulation to encapsulate different types of mRNA formats, like we use the SM102 to encapsulate the linear mRNA, and at the same time, we use the same LNP formulation to encapsulate saRNA. And we just find the in vivo biodistribution profile as totally different. For the linear mRNA group, the mRNA is mainly distributed or expressed in the liver, which is very typical phenomenon. But for the saRNA group, we found that even the LNP is mainly distributed in the liver. Their expression mainly distributed demonstrates very significant extrahepatic properties. And more interestingly, when we use different saRNA derived from distinct alpha viruses, we found that their expression profile is totally different from each other. So that gives us to hypothesize that might be the backbone, or the vectors themselves have some influence on the saRNA biodistribution properties. So for these findings, we also organized a preprint paper. Now the preprint version is available online. So if you're interested in this part, just feel free to check out. And I just attached the link and also the title of the paper here for your reference. So in addition to the vaccine development or immunization, or the interesting findings of the saRNA, the mRNA LNP can also be used for antibody discovery studies. So in this slide, I just shared a general workflow for the mRNA LNP-based antibody production from the very beginning of mRNA IVT and also LNP formulation to the animal model immunization and to the final antibody screening and validation. And on the right, we just show some data that we use different types of LNP formulation to encapsulate the human CCR9 mRNA. And we found that all of these three LNPs show very high serum titer. And we also compare that using different types of mRNA or different formats of mRNA and found that the saRNA has the higher serum titer level compared with other two formats of mRNA. So in this slide, we just give a full case study of saRNA LNP-based rapid immunization for human CCR9 monoclonal antibodies production. And in this case, just after one round of B-cell cloning, we got about 394 VAX-positive clones and narrowed down to 20 candidates after NGS sequencing. And finally, we got about 9 recombinant clones with very excellent EC50 values. Well, in addition to the vaccine and also antibody discovery applications, the mRNA LNP are also promising in cell therapy application fields. In this case study, we just compared two types of mRNA encapsulated in the SM102 LNP. One is the circular RNA, and the other one is linear mRNA for the CAR protein expression in primary T cells. And we found that the circular group just have better performance. Even after 72 hours, the circular RNA group has higher expression level, and the linear group just dropped very significantly. Well, then we go into the genome editing application fields. Gene editing is a very hot research area recently, and we found that the mRNA and LNP can also have some promising results in this field. When we use the LNP to simultaneously encapsulate both the Cas9 mRNA and sgRNA, we found that not only in vitro, we got a very good knockout efficiency. As you can see in 3T3 cells, we found that we got 76 to 78% knockout efficiency. And then we go into the in vivo animal study and find that we got about 64% knockout efficiency. Well, all of these case studies just show the potential of mRNA LNP in various kinds of application fields. So in the last, I would like to show some a little bit more about our targeted LNP. As I mentioned just now, in GenScript, we offer different types of targeted LNP. And in this slide, I just show some in vivo biodistribution data of these targeted LNP. As you can see, the CD3 antibody conjugated LNP, we found that in vivo, it has higher spleen expression level and the spleen expression ratio compared with the control group. For the control group, we just use the general SM102 LNP formulation. And for the GalNAc LNP and 20-DODAP LNP, there are two types of liver-enhanced targeted LNP. And in this data, we just found that both the GalNAc LNP and the 20-DODAP LNP has higher liver expression level compared with the SM102 LNP group. And the 50-DODAP LNP just show a very significant lung expression specificity compared with the control group. And also the 18PA, it has a specific spleen specificity in this case. And in addition to the tissue and organ level assays, we also did some in vivo cell specificity test. In this case, we just test the CD31 antibody conjugated LNP. Not only we found that in the tissue and organ biodistribution level, it has higher distribution or expression level in lung compared with the non-conjugated LNP. And also we found that when we obtain the CD31 positive cells from the mice lung. And we found that in those CD31 positive cells, the CD31 antibody conjugated LNP just has a significantly higher expression level compared with those unconjugated LNPs. Well, to summarize, all of these case studies I shared just now are enabled by our mRNA and LNP platforms in GenScript. So we can offer one-stop solutions from the gene synthesis to the plasmid preparation, and then to the IVT mRNA synthesis, and then to downstream lipid nanoparticle delivery system development. So we are looking forward to collaborating with all of you. And if you're interested, just feel free to reach out and we can discuss more about how we can help your research projects work smoothly. Well, thanks for your listening. That's all I want to share today. Open to the questions.
Thank you, Dr. Liu, for your insightful presentations. We have a few questions from the audience, and let's take some time to address them. So how do you conjugate targeting ligands to LNPs and ensure their activity?
Okay, that's a good question. Actually, we use some lipids with functional groups like the PAC lipids with maleimide, and then we will post-inserted these kind of lipids in our LNP surface, and then the LNP with those functional group can react with the antibody or peptides, those with functional groups on the ligands like CH group. And then we can do the conjugated LNP. And finally, we will conduct some QC items to test the binding efficiency, and we will do some downstream in vitro expression test to validate its performance. Yeah.
Yeah, okay. The next question, could you please elaborate on the potential of saRNA and circular RNA as a diagnostic target?
Yeah, that's a good question. Actually, for saRNA, usually we would use it for vaccine development, as I shared just now, because the saRNA has relatively high immunogenicity, and as it has long-term effects, so it's more suitable for the vaccine development. And for circular RNA, usually we will use it for cell therapy because it's a very suitable and welcome for those immunogenicity sensitive tests.
Yeah, thank you, Dr. Liu. And our next question, is GMP manufacturing available for LNP mRNA?
Usually in GenScript, we offer RUO grade and GMP-like grade mRNA and LNP services, but we do have GMP capacity in our sister company ProBio, so it's okay for GenScript.
Okay, and due to time constraints, we are taking just one more question. Do you have LNPs that target B cells specifically?
Well, that's a good question. Actually, currently in GenScript's R&D team, we don't have targeted LNP specifically targeting to B cells, but if we also can offer some customized LNP service. That means if the customer has some promising candidates or ligands like antibodies or peptides that can specifically target to B cells, they can give their ligands to us, and then we can do the conjugation on the LNP.
All right, got it. And thank you once again, Dr. Liu, and thank you to all our distinguished speakers for your outstanding presentation and discussions today. On behalf of GenScript Biotech, we sincerely appreciate your participation in today's webinar. We hope the insights shared today will support and inspire your future research and innovations. As the webinar concludes, a short 1-minute survey will pop up on your screen. We will appreciate your feedback. It helps us to continue to improve and tailor our future events to better meet your needs. Thank you again for joining us, and we look forward to seeing you at our future webinar. Wishing you a wonderful days ahead. Goodbye.